Hi everyone, my name is Sydney Vaughn, and today I will be discussing the complement system. I'm so excited. That is one of my favorite topics in Immuno. And before we get started, I just want to say that you know this information and you got this. Let's get started. So today we will be discussing the complement system. The complement system is an aspect of innate immunity, and the main goal is to eliminate pathogens. Now, there are three pathways. For today's purposes, we'll be focusing on the classical pathway. So to start, your antibody, which can either be IgM or IgG, is going to bind to your antigen. So you have antigen plus antibody means initiation. Okay, so now we have your C1 complex, which is composed of your C1q and your C1R2S2, forming your C1 complex. So how does this really work? Well, your C1q is going to bind to your antibody-antigen complex, which then causes your C1R2S2 to be activated. What does that mean? So first, we have your C1q binding to antibody plus antigen. Second, we have activation of C1R2S2. When this binding occurs in this first step, the first C1R gets activated, which then activates the C1R next to it, creating like a cascading effect, right? So then this C1S is activated, therefore activating this next C1S. So now your whole C1R2S2 complex is ready to go. Well, the next step is called cleavage. Cleavage just means to cut. You have your C1S that is now activated, and it notices this C4. So what's it going to do? The C1S is going to cut, these are scissors, sorry for my drawing abilities, but it's going to cut this C4 molecule in half, creating C4A and C4B. Well, the C4B notices that this C2 is just floating around. This is the C2. So it's going to bind. When the C4B and the C2 combine, another C1S is going to come. And what does the C1S like to do? It likes to cut. It's going to be a long arrow, so I'm just going to draw this C1S here. And it's going to cut this C2 molecule that we have here in half. And now we have C2A and C2B. Well, what happens next? Well, you have your C4B from before, and now you have the C2A that was just cleaved from the C2 protein, and they're going to combine to create the C3 convertase. And the C3 convertase can be written as C4B2A. C3 is going to be cut in half by your C3 convertase, forming C3A and C3B. C3B that was just cleaved, your C4B, and your C2A from your C3 convertase, making your C5 convertase. So that C3B, this is what this arrow means. That C3B from your C5 convertase notices a C5. So what does it want to do? More cutting. So your C5 is then going to be cut in half to form C5A and C5B. This C5B is going to embed itself in the cell membrane, coupled with your C6, your C7, your C8, and a whole bunch of C9s. And as you can see, this is in the cell membrane. and it generates a hole. This hole in the cell membrane allows for, that's what these little yellow means, cell lysis to occur, which goes back to our goal from the very beginning, which was eliminating the pathogen because the cell is lysing. So that, my friends, is a classical pathway. I hope it makes a little bit more sense, and if you have any questions, please feel free to email me.